Direction of non-template strand. 5-3. Direction of template strand. 3-5. RNA polymerase moves along which strand? Template. The RNA transcript is identical to which strand? Non-template. The protein sequence directionality is NC terminus. This element stabilizes incoming NTP by binding to the phosphate group. Magnesium. In RNA synthesis, the incoming NTP is attacked at the alpha phosphate by the blank of the growing RNA chain. RNA polymerase contains a negatively charged asp. How many RNA polymerases do prokaryotes have? One with multiple subunits. This prokaryotic RNA polymerase subunit is required for assembly of the enzyme, interacts with regulatory proteins, and is involved in catalysis. Alpha subunit. This prokaryotic RNA polymerase subunit is involved in NTP binding and catalysis, chain initiation, and elongation. Beta subunit. This prokaryotic RNA polymerase subunit binds to the DNA template. Beta prime subunit. This prokaryotic RNA polymerase subunit is required to restore denatured RNA polymerase in vitro to its fully functional form. Omega subunit. This prokaryotic RNA polymerase subunit directs the enzyme to the promoter. Sigma subunit. The 5 channels in the RNA polymerase are NTP uptake, RNA exit, non-template DNA, template DNA, and downstream DNA. The first step of transcription is binding of RNA polymerase hello enzyme at promoter sites. The second step of transcription is initiation of polymerization. The third step of transcription is chain elongation. The fourth step of transcription is chain termination. The position of the promoter region is -1. Transcription begins at what position? 1. The rate of DNA synthesis is 50 nucleotides per second. The elongation enzyme is core polymerases without sigma subunit. Elongation rate is 50 base pairs, slower in GC-rich regions. This enzyme must precede and follow polymerases to relieve supercoiling. TATA polymerase. The termination factor protein and an ATP-dependent helicase is Rho. The Rho-independent termination mechanism involves formation of this structure in the RNA transcript. Stem loop. What is required for Rho-dependent transcription termination? ATP. RNA polymerase I transcribes rRNA. RNA polymerase II transcribes mRNA. RNA polymerase III transcribes 5srRNA, tRNA, and other small RNA genes. Which two eukaryotic RNA polymerases recognize a single promoter? 1 and 3. The major assembly point for the proteins of the pre-initiation complexes of polymerase II. TATA box. DNA is unwound at which sequence? Initiator. What is responsible for recognizing and binding the TATA box sequence in DNA? TBP. Where does TBP bind in the DNA to unwind it? Minor groove. The 5' of mRNA is a 7-methylguanosine. The 5' of mRNA is attached in a 5-5 triphosphate linkage. Chemical group often found at the 3' position of the first and second nucleotides. Methyl. Synthesis of the 5' is carried out by enzymes tethered to the CTD of Pol II. This marks the completion of RNA polymerase II switch from transcription initiation to elongation. Capping enzyme binding to RNA PII's phosphorylated CTD. Capped mRNA are resistant to 5-exonucleolytic degradation. This identifies the translation start site. 5 cap. 2 enzymes involved in adding the poly-A tail in eukaryotes. Endonuclease, polyadenylate polymerase. Generation of the poly-A tail requires ATP. This protects mRNA from degradation. PABP. When can splicing occur? Either before or after cleavage and poly-A steps. Class of introns that are self-splicing, and require a guanine nucleoside or nucleotide factor. Group 1 introns. Class of introns that are self-splicing and form an intermediate branched lariat structure. Group 2 introns. Largest class of introns, requires spliceosome. Spliceosome introns. Introns found in terminus require ATP and endonuclease. ATP and endonuclease-dependent introns. The first step in self-splicing of group 1 introns is transesterification reaction. Splicing is mediated by blank in the spliceosome. snRNPs. snRNPs that participate in splicing have high blank content. Uridine. The spliceosome brings together pre-rna, snRNPs, and pre-rna binding proteins. These are processed by nucleotide removal, addition and modification. Terminus. tRNA molecules assume this shape. Clover leaf structure. Which end is processed first for tRNA? 5. What is added to the 3 end of tRNA? CCA. Transcriptoma. The sum of all the RNA molecules produced in a cell under a given set of conditions. How is transcription and DNA synthesis similar? Chemical mechanism, polarity, all 4 NTPs, Mg2 and use of a template. How is transcription different from replication? It does not require a primer and involves only emitted segments of a DNA molecule, no exonuclease. Activity. Also, only one DNA strand serves as a template for a particular RNA molecule. RNA is synthesized by what enzyme? DNA-dependent RNA polymerase. What does RNA polymerase require? All 4 ribonucleosides, DNA template, Mg2. How does RNA polymerase elongate the RNA strand? The 3' of the growing RNA chain and act as a nucleophile that attacks the alpha-phosphate of the incoming ribonucleoside triphosphate. How is the template strand copied? In 3-5 direction, anti-parallel to the new RNA strand. When does initiation occur? When RNA polymerase binds at specific DNA sequences called promoters. What forms briefly during elongation of transcription? Hybrid RNA-DNA double helix, transcription, bubble. How long is the hybrid RNA-DNA? 8 BP long. How many BP are kept unwound during transcription? 17 BP. What helps to relieve tension during strand rotation? TATA polymerases. Upstream, supercoil type. 5-side, negative. Downstream, supercoil type. 3-side, positive. Postulate of Jacob and Monod. Characterized mRNA. 4 properties: 1. Base composition reflect DNA. 2. Heterogeneous with respect to mass. 3. Able to associate with ribosomes. 4. High rate of turnover. Brenner, Jacob, Meselson Experiment. T2-phage. What is bacteriophage? T2. Virus that infect E. coli. After infection, T2 DNA determines protein synthesis. 0. Info. RNA polymerase rate of nucleotide addition: 50 to 90 nucleotides sec. How many subunits does E. coli RNA polym have? 5 core subunits. Sigma subunit function. Binds to the core and directs the enzyme to the binding site on DNA. Initiator factor. Most common sigma subunit: sigma 70. What activity does RNA polym lack? Proofreading 3-5 exonuclease active site. Rate of error for transcription: 1 error for every 10-4 to 10-5 ribonucleotides. The RNA transcript is similar to which DNA strand? The DNA non-template strand. Where does RNA synthesis begin? At promoter sites. Promoter sites locations: -10 TATA, -35 TTA. Consensus sequence. Certain nucleotides that are particularly common at each position. Efficiency of RNA polymerase. Determined by consensus sequences, spacing between and the distance from the transcription start site. Pathway of transcription initiation: closed complex formation, open complex formation, start of RNA synthesis, promoter clearance. E. coli protein that activates transcription. CAMP receptor protein increases the transcription of genes coding for enzymes that metabolize sugars other than glucose when cells are grown in absence of glucose. Repressors: proteins that block the synthesis of RNA at specific genes. E. coli termination signals. Rho factor protein recognizes and binds to C-rich regions, advances in 5'3" direction to bubble, unwinds the helix and releases nascent mRNA, Rho-independent, forms hairpin structures before the projected end, conserved string of 3A residues in template strand. How many types of polymerase in eukaryotic cells? 3 types. Poly. Synthesis of pre-ribosomal RNA. Pol II. Synthesis of mRNA and specialized RNAs can recognize thousands of promoters that vary in sequence. Has TATA box and INR sequence at 1. Pol3. Mixed-RNAs, 5SR RNA and OTHR small specialized RNAs. Spiegelman Experiment. T2 mRNA labeled with 32P, T2 DNA labeled with 3H. Mix, heat and cool. This demonstrates that RNA has complementary bases. Denaturing needs to happen for comigration and hybrid formation. Alpha 2 subunit function. Assembly and activation of enzyme by regulatory proteins. Beta. Binds and TPs and interacts with sigma. Beta. Binds DNA. Sigma. Recognizes promoter sequence of DNA. How many BP are unwound to form open promoter complex? 12 pairs. Footprinting is used to identify specific binding site of a repressor, polymerase or other protein on the DNA. Pol2 is most sensitive to what? Alpha-amanitin, octopeptide from Amanita phalloides. It is an inhibitor for Pol2. Enhancers are also known as upstream activation sequences. Transcription factors. Recognize and initiate transcription at specific promoter sequences. What are some transcription factors? TFIIA and TFIIC for RNA Pol3 bind to specific sequences within the coding region.